As you age, your cells accumulate damage, but not all cells are affected equally. Some cells, like neurons, are rarely replaced, while others, like skin cells, regenerate frequently. This disparity is crucial. Neurons in your brain can last over a century, storing a lifetime of memories. Meanwhile, skin cells turn over roughly every month, maintaining your protective barrier. Surprisingly, this cellular replacement isn't flawless. Each division introduces tiny errors in DNA, akin to making copies of copies. By age 60, each cell may harbor thousands of these small mistakes, setting the stage for aging and disease. Why does this happen, and what triggers these changes inside you? Deep within you, telomeres, the protective caps on your chromosomes, shorten with each cell division. Think of them like the plastic tips on shoelaces. Their length determines your cell's lifespan. In a newborn, telomeres measure about 8,000 base pairs. By the time you reach 80, they can dwindle to around 1,500. When telomeres become too short, cells enter senescence, a state where they stop dividing. This might sound counterproductive, but it prevents potential cancerous growths. However, these senescent cells can release inflammatory signals, disrupting tissue function and contributing to age-related decline. What incites this cellular self-sabotage, and why can't it be avoided? Your mitochondria, the powerhouses of your cells, also play a significant role in aging. They generate energy by producing ATP, the currency of cellular power. However, this process isn't entirely efficient. Reactive oxygen species ROS are byproducts, acting like molecular sparks that can damage DNA, proteins, and lipids. Over time, ROS accumulation leads to oxidative stress, which impairs cellular function. Interestingly, your body has evolved antioxidant defenses like glutathione to combat these radicals. But with age, these defenses weaken, allowing more damage to occur. Why do mitochondria generate these harmful sparks, and how do they accelerate cellular aging? Inside you, the immune system's efficiency declines with age, a phenomenon known as immunosenescence. In a 2018 study, researchers observed that T cells, the soldiers of your immune defense, become less responsive over the decades. By age 70, their ability to combat new infections drops significantly. This decline isn't just due to fewer cells, it's also about diminished communication. Cytokines, the signaling molecules your immune cells use, don't transmit messages as effectively. This impaired dialogue leaves you more vulnerable to diseases. What triggers this breakdown in immunological conversation, and can it be mitigated? The extracellular matrix, a supportive network surrounding your cells, undergoes changes as you age. Collagen, a primary component, provides structural integrity to tissues. But from your 20s, collagen production decreases by about 1% annually. By age 60, your skin can lose up to 30% of its collagen. These losses contribute to wrinkles and sagging. The matrix also affects your joints. Cartilage, which cushions bones, wears down over time, leading to stiffness and pain. Interestingly, this breakdown isn't entirely from wear and tear. Enzymes like collagenase actively degrade these proteins, raising the question, why does your body dismantle its own support? Inside your cells, DNA repair mechanisms work tirelessly to correct errors in the genetic code. One such mechanism, nucleotide excision repair, identifies and cuts out damaged DNA segments. Enzymes then fill the gaps with correct nucleotides. However, this process isn't foolproof. As you age, repair efficiency declines, leading to an accumulation of DNA damage. A 2020 study revealed that repair enzyme activity can decrease by up to 40% in aged cells. This decline contributes to mutations. Interestingly, your cells prioritize repairing genes crucial for survival, a strategy to maintain essential functions. Yet, why can't these mechanisms fully prevent the ravages of time? Autophagy, the process by which your cells clean out damaged components, is another crucial mechanism. Lysosomes, cellular organelles filled with enzymes, engulf and digest damaged proteins and organelles. This recycling process is vital for cellular health, but it diminishes with age. Research published in 2019 indicates that autophagic activity can decline by 50% in aged tissues. This reduction leads to an accumulation of cellular debris, impairing function. Autophagy even contributes to neurodegenerative diseases as protein aggregates build up in the brain. Why does this self-cleaning system falter, and how does its decline impact aging? Your cells communicate through signaling pathways, intricate networks that control growth, division, and death. The insulin signaling pathway, for instance, regulates metabolism and longevity. However, with age, sensitivity to insulin decreases, a condition known as insulin resistance. This resistance can lead to elevated blood sugar levels. A study from 2017 found that insulin signaling diminishes by about 30% in aged tissues. This disruption affects cellular energy balance and can accelerate aging. Surprisingly, interventions like calorie restriction have been shown to enhance insulin sensitivity, suggesting a possible modulation of aging processes. But why does this pathway become resistant over time? Epigenetics, the study of changes in gene expression without altering DNA sequence, reveals another layer of aging complexity. Methylation, the addition of chemical groups to DNA, can silence or activate genes. Over time, methylation patterns shift, altering gene activity. A fascinating 2018 study demonstrated that DNA methylation changes are among the most reliable biomarkers of biological age. These shifts can affect genes involved in inflammation and repair. As a result, the epigenetic landscape of your cells becomes noisier, potentially leading to dysfunction. What causes these epigenetic alterations, and why are they so closely tied to the aging process? Protein homeostasis, the balance of protein synthesis and degradation, is essential for cellular health. Chaperone proteins assist in folding new proteins, ensuring they achieve their correct structure. However, with age, this balance is disrupted. Misfolded proteins can accumulate, forming aggregates that disrupt cell function. Interestingly, a 2021 investigation found that the efficiency of chaperone proteins decreases by approximately 20% in older cells. This inefficiency contributes to diseases like Alzheimer's, where misfolded proteins are a hallmark. Why does the protein maintenance machinery falter, and how does it affect cellular longevity? Under extreme conditions, your cells can experience what's known as cellular senescence. These cells stop dividing but don't die. Instead, they linger, releasing inflammatory signals. While this process prevents cancer by halting the division of damaged cells, it can create problems when senescent cells accumulate. In your joints, for example, they contribute to arthritis. In 2019, researchers found that removing senescent cells in mice extended their lifespan by up to 35%. This suggests a profound impact on aging. Yet, how do these zombie cells manage to disrupt the entire cellular neighborhood, and is there a way to safely eliminate them from your body? When your body's repair systems falter, diseases can emerge. In the context of cardiovascular health, arterial stiffness is a prime example. Collagen and elastin, proteins that give blood vessels flexibility, degrade over time. This degradation increases blood pressure and heart disease risk. A 2020 study highlighted that arterial stiffness can rise by 50% between the ages of 40 and 70. But it's not just physical wear and tear. Cellular signals that promote inflammation contribute to this stiffening. How does this cascade of biochemical messages lead to such profound changes in your cardiovascular system, and can interventions alter this trajectory? At the cellular edge, cancer represents a breakdown in regulatory mechanisms. Normally, tumor suppressor genes and oncogenes balance each other, preventing uncontrolled growth. With age, mutations accumulate, disrupting this balance. Incredibly, by age 70, some studies estimate a person may carry several hundred trillion mutations across their cells. Yet only a fraction lead to cancer. This suggests complex layers of control within you. Immune surveillance also plays a role, identifying and destroying emerging cancer cells. Why do some mutations slip through these safeguards, and how does aging tip the scales towards malignancy? Your brain, a marvel of complexity, isn't immune to aging's effects. With time, the hippocampus, critical for memory, can shrink. This shrinkage is linked to cognitive decline. A 2018 study found that the hippocampus can lose up to 20% of its volume by age 80. But it's not just about cell loss. Synaptic connections, the communication bridges between neurons, also decline. Interestingly, some parts of your brain can compensate by rewiring themselves, a process called neuroplasticity. How do these dynamic changes maintain function despite structural losses, and can we harness this adaptability to combat age-related decline? In extreme cases, the Hayflick limit reveals the limits of cellular division. Most of your cells can divide roughly 50 times before senescence. This limit is driven by telomere shortening. However, certain cells like cancer cells bypass this limit, achieving what's known as cellular immortality. They activate telomerase, an enzyme that rebuilds telomeres. In 2021, research showed that nearly 90% of tumors exploit this mechanism. This reveals a paradox. While telomere maintenance could theoretically extend life, it also opens the door to cancer. The interplay between cellular aging and immortality challenges our understanding of life itself.